As you might guess from this first title, I'm not going to be really heavy on the whole microbiology or the molecular biology. My background is very much from a process perspective, so that's the process engineering and the chemistry, environmental fate behind that. So I thought I'd start with giving a bit of an overview of what I do. So the first thing is, I kind of look at what's called bioprocessing. So that can be any process that has a biological input or an entity. So a wastewater treatment plant, a river system, a farm setup, anything where you've got a nice microbial consortium. I might not know a lot about them, but I know from a process perspective. And I'm interested in looking at the efficiency of the process and also resource resilience. And the resilience word is because you can then look at how, if you have a shock to the system, how can that process actually deal with it? So when you're talking about a resource, that could be in terms of the quality of the water. It can also be about the things that are in water, so potential pollutants that you can then recover and become valuable. So I have a particular interest in pharmaceuticals, steroids, and also comes along with water, with metals as well. So it's very driven by what the water industry are interested in and what they have to do in the funding. And I really look at having to develop analytics, looking at the fate, and then process technologies as well. Now, a particular interest of mine is the complexity, and Dov was mentioning that before. If you take a wastewater treatment plant, there's a lot of different factors there. So how can I look at best removing that pharmaceutical of interest, for instance? The idea being that that process wants to deliver that product at a particular specification. So that could be clean water. I also look in manufacturing environments when you look at waste or biomass and extracting materials of value. But you often have a very variable starting point. Your feedstock is variable. So how can your process deal with that variability and the complexity while still delivering that product on spec? So I thought I'd start with what we all know. Everything started back in the 30s. It was mainly for human use, and then they started looking, because we were looking at more intensive processing of animals in a very big farm environment, it led to having these antimicrobials in veterinary use. Now, if you're going to have exposure of antimicrobials to humans or animals, you're invariably going to have exposure to the environment. If for no other reason, it will go through you and it will go through the animal. So this is where the concept of that One Health came from, that you need to look at the humans, the animals, and the environment as being intimately linked. So what this shows is, again, those that were first deployed, and then when you're having the resistance that happened. And what is clear is that that spread of resistance is really being outpaced by the rate of our drug development. So we all know the issues that are related with this antimicrobial resistance issue. So I thought I'd start with, because my talk's about the outside environment, defining some example outside environments. Out of interest, how many people have ever been to a wastewater treatment plant? Yes. How many have ever lived in a wastewater treatment plant? Yes. Okay, I'm going to have to talk to you later. So starting off with a farm, same thing with the farm setup. You can be looking at cows, pigs, chickens, whatever it might be. And to give those two pictures there, straight away, a cow farm, for instance, is not a one-size-fits-all. And it's not just a case of it being dairy or meat. It can be outside, it can be inside, it can be a mix. The downside picture is actually, it's a farm in the States where that particular farm is processing 32,000 cows. And the circular bit, if this works, no, that circular bit there is where all the actual, the muck goes, so where all the manure and the slurry is actually going. So you can see it's quite a nice, intensive process if you just want to get a whole load of cows through the system. So when you're talking about the farm, you've got the cows, you've got the slurry tank, you've got the lagoon, you've got the manure that might be going through drying and then might go to land. So you have a lot of different stages. So which stage is of most relevance? Is the one stage of most relevance? How do they interact with each other? Going from the gut to the tank to the lagoon, how do they impact and influence? And this also talks and goes further than the farm environment, is what's the influence of waste management practices. And this is very important and will continue to be so, because this is where you can have a real monetary thing as well if you're looking at antimicrobial resistance as an emerging pollutant, where it ends up being a legislative issue potentially. And it's where it's gone from the pharmaceutical perspective in the water industry. So really where we are right now is that further information is required to understand AMR selection and also the spread of it in the outside environment. So these are some of the other outside environments. Think of your river system. So if you take an antibiotic, the reason why the antibiotic has been in the river is because most wastewater treatment plants, once treated their wastewater, will release it into a river system. And they were never designed to remove these sorts of chemicals. So they enter river systems, and straight away, if you think of the fate of that antibiotic pollutant, and that might not necessarily be the antibiotic, that can therefore then be the resistant gene, the resistant bacteria, you're looking at a lot of different relationships within that one outside system. What's a nice one, potentially, is particulates. So particulates, antibiotics, if they've passed through the wastewater treatment plant, they're mostly going to quite fancy particulates. And a lot of microorganisms like it as well because it's very nice for just shielding them from UV and for other things. So are particulates a site for selecting or spreading AMR? Do we really know that? What other aspects within that river system might be the case? How might they transport? How might they work between the liquid and the solid phase? You've also then got the aquaculture where we're growing more and more into, so that's very intensive, stick a lot of fish somewhere, but you have that flow-through system. The reason why you have the flow-through system is because just like with a lot of farms now, we're doping our fishies or our cows or whatever with low levels of antibiotics. And if it's got that flow-through system, it then stops the food and the high COD, so chemical oxygen demands, but it also will allow those antibiotics to move through and outside of that circle. You've got your crops, and the application or not to manure. This is another outside environment. You have your wastewater treatment plant, which I'll talk a little bit more as a case example environment. And you also have landfill. So these are just, I'm sure it's not everything, but just example outside environments. So the landfill is where you have inappropriate disposal of antibiotics. So I'm sure you all know that you're supposed to be giving your antibiotics, if you do not finish them, to a GP or a hospital, and it will go and become incinerated. A lot of people still put it down a bin, or they'll put it down the toilet. If it goes down the toilet, it will go to a wastewater treatment plant. If it goes into a bin, it will go to landfill. In Nottingham, most people goes to an incinerator, but that's a bit different, Nottingham. But most places will go to a bin and into landfill. Now landfill, first of all, you've got your vectors, and then you have your collection of your leachate, which is basically the landfill wastewater, which will be collected every so often and tankered to a municipal wastewater treatment plant. So it's still entering that wastewater treatment plant. And a nice, lovely concentrated dose that hasn't gone through the human. So straight away, your routes with which antibiotics enter a system, and it's not just the antibiotics, but also the antibiotic resistant bacteria or genes as well, whether they're already there or whether they're then being selected. And it's clear that if you're talking about an outside environment, defining the boundary is not easy because they are not closed systems. They can be linked in with inside, with humans and animals, indeed the manufacturing process or other outside environments. And this is when, and Dov gave an example of this, so when you start looking at environmental relationships. And there's a lot of work looking at this at the moment because it helps to identify the boundaries and how things are actually selected and transferred within a system or between systems. So if you take this as an example, normally we will have indirect water reuse. So what that means, it will go through an environmental compartment. So if you have your industry or household, it goes to a wastewater treatment plant, the aqueous phase will go to a river, and then it will go usually to a drinking water treatment plant or you'll bathe in it. That's indirect. However, we're starting to see changes in the way that we process and reuse our water and that we're moving towards direct water reuse. And what that means, if you take Australia, which I clearly can't spell, that has different treatment classes. So if you treat it to a class A, and that means you then have filtration and UV, it is then going to be fit for car washing. So it leaves that wastewater treatment plant, they don't call it that, they actually call it water reclamation plants over there. It leaves that plant and it will go directly to a tap outside of a house in a community, and they will then use that for car washing. And because obviously with the spray, that's why you have that higher level of treatment. So you've got much closer contact then, you've got a different route of which it can apply to humans. There is also potentially, with climate change being a driver, because Australia and certain parts of the States are very having a lot of problems with water. So what I've done with this is just given a couple of examples of some papers. No means does this mean that this is the best paper, it's just an example of what some of the work is going on. And it's very similar to what's happening with the whole pharmaceutical issue that started 20 years ago now, almost to the year. So here's a paper where they're looking at reclaimed water irrigation. So they've taken a wastewater, they have reused it, and they're using it for irrigation, in this case to a park. And they looked at the profile of antibiotic-resistant genes, and they're able to find a correlation with transphosphate abundances, and they were detecting it among different park soil samples. So again, another route with which you can have contact to humans, to animals, and just showing the complexity of that outside environment and how they overlap and interact with each other. This is something by the United States Department of Agriculture, who are doing a lot of work on the ecosystem, obviously especially with agriculture and animals. And they were very clear that they didn't want this uni-bilateral approach, which is why they've put a pretty little circle. And they looked at direct sources, indirect sources, and it's clearly said that no source or activity within a circle is independent of the other, which is clear, what we expect. And it also highlights that travel, that global and national and local context. Now this is where we are end of last year with what the Department of Health did. It was actually 4 governmental agencies. They've got, for those who haven't seen this yet, they've got about 6 or 7 different systems maps, and this is the overview one. So they've got the environment, animals, human, food, and then it gives an idea of the complexity, different things. So in pink you've got like the environment, you've got wildlife, you've got humans. It then shows different sources, different influences, in a very nice way of how it all relates, and how if you're talking about that outside environment, where are your boundary conditions? How do they relate onto others? Oh, it's like an eyesight test. So when you look at the motivations for considering antimicrobial resistance in the outside environment, our first approach and our primary approach has been about the impact on human health and also on animals, which we might then eat. So there's several routes with that. So some people have been looking at occupational exposure with those who work with piggies. Others have been looking at manure fertilisation and how that then impacts on soil and vegetables at harvest. And it's always worth just looking at, you know, certain cases like the bottom one, what are the concentrations, because it was a laboratory setup, what are the concentrations with which they dope the antibiotics to then encourage that or drive that. There's also another aspect, and this is more from a process perspective, of whether there is a duty of care. So this is a sort of a common law approach, and this is something that happened to the water industry. They got caught with it about 15 years ago to do with pharmaceuticals. And what this means is the water industry, which for those who do not know, in England and Wales, we ignore Scotland and Northern Ireland, I have no idea why. In England and Wales, there are 23 water companies, and they are responsible for our drinking water and for our wastewater treatment. So we're in the Severn Trent land. Okay, Severn Trent are the second largest, and they hold the highest environmental range quality guidelines. So they are required as an industry to protect the quality of water resources and the organisms within. It's one of what their requirements. And this just gives you a basic view of how we're seeing antibiotics, antibiotic-resistant genes, bacterial, what else, going into that wastewater treatment system. And I'm just going to give a couple of examples about that as a case study outside environment. So as I said, they're responsible for this wastewater treatment plant. which is primarily made up of two main stages, primary and then your biological secondary stage. Now obviously the pharmaceuticals, antibiotics, go through us, go through the toilet and enter. They can also go through the industrial system as well, because even if you have a manufacturer manufacturing antibiotics, they'll often have their own wastewater treatment system, or they might then treat it to a certain level and then put it to this municipal, which means community, wastewater treatment plant. And then it's that urban water cycle, goes to receiving waters, and then it can literally be a couple of yards down the river, you've then got your drinking water treatment plant. So you have that lovely cycle. Now, the wastewater treatment plant was never designed to remove things like antibiotics. And for the same reason, it has not been designed to remove things related to antimicrobial resistance. So what is happening, and I'll mention for those who might not be aware, what is happening and started 15, 20 years ago with antibiotics and pharmaceuticals and steroids seems to be exactly the same case now from the wastewater treatment perspective and the outside environment with antimicrobial resistance. So there might be lessons that we can learn, analytics being one of them. If you don't have the right analytics, then you might not be getting the full story. And that was certainly the case with antibiotics. So if you look at wastewater treatment plants, and I've said this, back in the mid-90s, they started seeing these things about dodgy fish and cancer, and they were genetically male, but they had female physiology and beta-gelatin and things like that. And this prompted the water industry and therefore the research community to start looking at why this is the case. And it started with steroids and certain industrial chemicals, which have since been banned, and it's moved on to pharmaceuticals. Now, the interest in these pollutants has meant that we're actually at a nice advantage here when we're looking at the issue of antimicrobial resistance, because we have a lot of literature about the prevalence of antibiotics within and across a wastewater treatment plant, as well as the receiving waters, as well as their fate and their removal, so looking at the technology perspectives. And where the water industry currently are now is they're at a 200 million investment. The last 140 started in April this year. And they're looking, it started off with why does their effluent adversely affect fish, and they're looking at ways and how they might mitigate the concentrations of antibiotics and other pharmaceuticals, as well as metals, from their effluent before it goes into the receiving environment. So this has meant, if you look back, a lot of changes. Analytics, the analytics that was being done even 10 years ago was not telling us the full story. So what we've had to do is look at newer analytics to track metabolism, because we excrete them as conjugates. They might or might not be deconjugated depending on what the conjugate group is. You then got all sorts of funny metabolites going on. It's great fun. And you've got partitioning between different levels. It's a really nice, interesting thing to try and work out what's happening to them. And how does that impact of antimicrobial resistance? If you have conjugates, is that going to be a driving force? It's also been looking at evaluation and also development of technologies, so individual unit processes that might be able to remove these pollutants effectively and also, importantly, cost effectively. But an example here, this is back from 2005 to 2010 is when they did it. They looked at several different technologies and they're finding costs, so GAC, granular activated carbon, quite standard, just cost prohibitive. So going back to antimicrobial resistance issue, what does this mean? Are we going to have to be looking at process technologies? Well, before we do that, we need to first understand what is happening in each of these outside environments. What is the prevalence? What is the fate and behaviour? What are the things that drive it? So is this really our new wastewater problem? And are antibiotic-resistant genes and the bacteria the new pollutants? So this gives a bit more of an example of an activated sludge process. I love this thing. There's a bloke called from Environment Canada who refers to this sort of stuff as Bacterial Bordellos. I think it's great. So as I said, the activated sludge process, you've got that nice aeration tank. After primary sedimentation, you then put it into a system where there's a lot of microorganisms, and there's so much you can actually see the flocks. And that allows them to then use the wastewater as a nutrient, as a food source. So it's removing the pollutants because it's acting as its food source. And then you clarify it out. So it is a biological process. Another one is an attached. So this is an opportunity for antibiotics to degrade, to attach to the flock, to pass through the unit process, perhaps looking at metabolism as well with that. How is that impacting? We've done so much work on this from just an antibiotic perspective, but we haven't done it from the antimicrobial resistance perspective. And it's only now that we're starting to look at it more. Now, this paper by Yang, there's some really nice stuff coming out from China. This just gives an example of them looking at antibiotic-resistant genes. And they've looked at influent and effluent, so the wastewater sides. They looked at activated sludge, so the flock, and then they looked at the ADS, which is the anaerobic digestion sludge. And it shows here that the different ARGs, they change as they go through the wastewater treatment plant, and they're also far better removed in the liquid versus the solid phase. Could this be related to antibiotics? What might be the other reasons? Metals like solids. So it shows here as well that a wastewater treatment plant might be releasing antimicrobial resistance into the receiving environment, but it also is receiving it. Do we know enough yet as to how much is going into that system and how much is that actually producing in itself? And why does the profile of ARGs vary? What's the reasons for that? Another example here is with not just antibiotics, but also heavy metals. And they found some correlation with a particular one. So it's showing that there is some complexity here, and what factors should we be looking at? And it's showing also that you've got the analytics, you've got the process engineering, you've got the microbiology, you've got those range of different disciplines coming together. This one is looking at your class 1 resistance integrons, and this looked at hospital effluents. So this was actually in a laboratory setup, and it went straight into a conventional activated sludge system, and they were looking at how that influences. So it shows that work is starting to be done, but there's still a lot to do. We still need to be making it a bit more joined up, and what does it mean? And also, it's not just as easy. So if you talk about an activated sludge plant, the two on that side are called diffuse, air bubbles from the bottom. The two on this side are called mechanical. So you're throwing the air up, turning it around, or the sludge up, turning it around and letting it come down. This has a lot of different in terms of spray, vectors, a whole load of other operational parameters as well. So one size does not fit all, and this is just a conventional activated sludge plant. We're now going into a lot more advanced treatment technologies now for activated sludge. So if you talk about activated sludge, what are the different variations and how do they impact on selectivity? And if you have an operational failure, so you can see here the little blank spots is where you haven't got diffuse or you haven't got the mechanical stirring. Or if you have design, so these are just some advanced design, and over the other end there in the green is showing this is actually a poor design of that particular mechanical. How does this all impact? There was a few years ago, they did a nice paper on Tamiflu, but they just took it from the antibiotic prevalence perspective and removal. And then this is looking at other treatment technologies. So in this case, it's UV and chlorination, and looking at horizontal transfer. And they found distinct differences whether you use UV and chlorination being more of an issue. What is worth noting is that this is in the States. Here in England and Wales, we do not use chlorination. So you're going to have different impacts depending on where you are geographically. So which treatment technologies are currently effective, or which is the most effective? How are they combined? Which are the ones that are actually promoting this selection on this spread? We really don't know enough at the moment. And it's not just the complexity, it's also the granularity. So some of the work that's going on is what level of detail is appropriate? How much should we know? Are we going down to enough detail? Are we going to too much detail? So if we're looking at horizontal transmission, should we know the different types? How will that help us? Wang, for instance, identified 2 dominant mechanisms in the reclaimed water irrigation. So is this information useful? Does it have the potential to be useful? But there are cost implications. The more you look and search and analyse for things, then the more cost is going to be. So to reduce the risk of antimicrobial resistance, you need to limit the selection of resistance and the spread of those resistant bacteria in those genes. What else? How can we achieve this is the first thing. And the second one, and this is who's going to pay? Who is the polluter? Is it those who overprescribe? Is it those who use it as a management tool, for instance, in farming? Is it the user in terms of the consumer at inappropriate disposal? The extended producer responsibility, so like the polluter pays, so the manufacturers. So a lot of manufacturers are doing things like PI, pharmaceuticals in the environment. Again, it's driven from removing these pharmaceuticals in terms of the pharmaceutical, but it has an impact on antimicrobial resistance. Is it going to follow the case of what's happened with the bioactive chemicals and endocrine disruptors, where it has been now the responsibility of the water industry? The reason why these sort of things are worth knowing is because it's all very well researching stuff, but if this is a problem, we need to work out how we're actually going to deal with it and who is going to pay for it. Or I'm just very hard-nosed. So just to finish off here, in 20 years, when they first start seeing those dodgy fish, you now have the Water Framework Directive, which has a watch list of emerging pollutants, which is a precursor to the EU priority list. If it's defined as an emerging pollutant, so things like estradiol, diclofenac, ethinyl estradiol are, then it means it will start conforming to quite a few different things. Are we looking and moving towards that? Maybe not today, maybe in a few years, because that has huge implications. It can potentially have implications on REACH, so registration, evaluation, authorization, and restriction of chemicals. Could it be a category of potential substances of very high concern, just like what is similar to bioactive chemicals or endocrine disruptors, which are seen is in a separate category? And I just wanted to finish with sort of communicating what we do and sometimes the media and challenges. There is stuff documented last year, found some really interesting stuff of what they're doing within a wastewater treatment plant. It's not necessarily the wastewater treatment plant that is the problem. We don't know enough to say that. It clearly is receiving resistant genes, resistant bacteria. Is it then actually producing more? What's going on? So when we communicate what we do, we have to ensure that we're telling the story, or at least the media is clear on what we're trying to say and whose fault or who is the polluter. Do we really know enough yet? Same thing when we talk to the public. This is something from a couple of years ago that was something that came out that said, well, you don't really need to take your drugs back. You can just dump them down the bin. Whereas the water industry is saying you do not do that. It's a huge, huge no-no. So again, it's making that clear message as a community to do that. And as I've gone through this, I've kind of done it as questions, because I find that there's a lot that we still need to be working on that is quite fun and challenging to look at. So do we consider it as an emerging pollutant? What do we need to know in order to define it as that? Will it help us? What sites do we monitor? So what sentinel sites should we monitor? And how do we define that site boundary and the interactions? How can we reduce the risk? Is it an educational answer, an engineering, an evolutionary one? Is it a mixture?